Hi, can you hear me?
Loud and clear. There you go.
Okay, I'm gonna just turn off my video and minimize because I'm at home. We had a big windstorm here. Anyway, um, yeah. So thank you so much for having me. Excited to be here, and I'm gonna talk about life detection in subglacial environments. This image you see here is of Olivia Vinnis, a graduate student, and myself actually sampling below a glacier, and that is definitely the ideal, but that is not how one really in the real world gets to sample subglacial environments in most glaciated systems. And I could drone on about this topic forever, but I'm going to try to keep it focused to the work that we did at JERP this past season and what we did in part of this life detection project that was funded by NASA. So, kind of relating it to analog environments. Okay. All right, you know, there's so many experts in the room on this, but when we think about subglacial environments, there is a ton of unknowns, and most of that is related to the hydrogeology of these systems. Renee just laid that out really well, even for North Basin, and these are important things to understand because of obviously the potential influence on ice sheet dynamics. But also on how that changes the chemistry that occurs below ice, as well as what happens to that water when it drains out. And this is where microbial ecology really steps in because it's the microorganisms that are inhabiting these environments that play a large role in mediating some of the chemical reactions that occur, changing the solute concentration of the liquid in the bed environment, and generating gases that may then subsequently flux to whatever your proglacial system is. And in order to understand these biological systems, we really do need direct exploration. This is not something that we can do well remotely. And it's important to understand that microbes inhabit all realms of the cryosphere, so all parts of the glacier. Folks who work at JERP understand well the surface component because they probably see some of the snow algae that comes about in the summer at the surface there. But microbes persist throughout the end glacial realm and really find a nice habitat in the subglacial realm as well. And so these features, like snow algae, can be visible from a large scale. Sometimes you can even remotely sense these large blooms of photosynthetic algae at the surface. But mostly they operate on the microscopic scale. And so here, these little green and red dots that you see are microbes that are actually growing and sticking to sediment grains within ice cores collected near the surface of a glacier, so in criconite holes. And one of the things about life in ice is that it lends itself well to how we maybe conceptualize the potential for life beyond Earth. And recent discoveries of numerous ocean worlds, even in our solar system, have kind of expanded what we think of as potentially habitable. And so the universe is a really cold place, and we have these moons of Saturn and Jupiter that harbor liquid oceans. There's growing evidence for potential subsurface liquid brines on Mars, but even in the absence of those, there are ice sheets, water ice sheets, as well as CO2 ice sheets on Mars. And so subglacial environments in the cryosphere in general offer really good analog environments for us to kind of think about how we might sample these places. And so that we know, understanding where to sample and how to sample happens now. The toolkit is enormous. There's numerous geophysical techniques that folks here in the room are experts on. But again, it's this direct access to the bed that's really critical for not only ground truthing what some of the remote sensing and modeling is doing, but also, in order to sample and understand the microbial communities that are present. I'm looking at the room and I don't see, are my slides advancing?
Yep, they are.
Okay, I think the Peter Strand one is just stuck on the Ocean World slide, so maybe that's not an accurate depiction. Okay, Whatever. I'll just keep talking. Sorry, guys. So the what I'm going to talk about that we did at Chirp is part of the NASA funded Peace Star program that doesn't exist anymore, but it was this cool idea where you could do planetary science and test technology in an analog environment. So get some really good terrestrial science done as well. And Juno was a great opportunity for us to test cryobots and propose kind of like this analog mission, like what would we do if we were on an ocean world, and use the Juno ice field as an analog for all of those operations. And so this enabled us to not only achieve engineering goals, like how to advance the robotics for this kind of subglacial access, but also to test terrestrial science goals. And in this case, it was to look at the life that exists within the Juno ice field. And so parts of this mission were, the radar work that Renee and Seth were leading as part of Orca is exactly what missions like Europa Clipper are conducting right now on Europa, in ocean world of Jupiter, ocean moon of Jupiter. There were components of this project that were designed to communicate on the timescale of delay that you would get if you were on Europa sending information back. We were using a cryobot to melt through in 2023. Jake and others helped us drill with a hot water drill. And then in 2025, we went back with a cryobot called the Ice Diver. And this is where we did that sampling and analysis component. And so cryobots are just a fancy term for melt probes. This is long tested technology that's been used throughout the cryosphere, modeled after what the original Filbert probe was. And it's basically just a probe with a copper tip that can penetrate ice by pumping some electrical current down into heat. And some of the more modern probes have differential heating, so you can do a little bit of control and navigation. The newer probes, like the Ice Diver that we used, has a lot more payload capacity and enables you to kind of target in situ sampling. And so, yeah, here's just a figure showing this concept design. Obviously, the deeper you go, the bigger your probe is. And so, our 2025 Ice Diver was a little bit taller than your average person. And none of these have been used yet on NASA missions, but there are folks who think that like these are the future. And I think regardless of their potential use for NASA, I think these are really powerful tools for cryosphere exploration because they do allow much more rapid access to the bed than drilling. And they allow a cleaner, more streamlined access to the bed than hot water drilling. So the cryobot ice diver that we used had a variety of instrumentation for control and measurement. But the unique thing about this one from our perspective was the inclusion of what was called a bladder pump. And so the reservoir or the payload section, we were able to put a sampling device. And in the past, we had bags that would collect discrete samples of about 150 mils of meltwater, which is not a lot. And this bladder pump allowed us to continuously pump water throughout and gain a profile. So we were able to collect water throughout the depth of the glacier as we went towards the bed. I don't need to go over this because Renee did an amazing job, but her and Seth and their team helped us select where to drill. Again, we were targeting some subglacial liquid reservoir. And to do this, we established what we called Space Camp at North Basin. Here's Coastal installing some of our equipment, and the dome tents are going up. Much of the work I'm going to talk about, like this hot off the press data, is from Olivia Venice, who is a PhD student in my group who led this work. And there's our biodome tent going up. I don't know. I mean, we could have discussions about the efficacy of these dome tents because we did lose them. We caught them again, but we did lose them during that field season. Um, so the main goal of the biodome was to detect the presence of life throughout the glacier and in the basal reservoir. So we took our tent and set it up like a microbiological laboratory. And so here you can see we have a variety of instruments. We try to keep it as clean as possible, and we use this setup strictly for microbiological analyses. Our goal was to look for biological signals or biologically suggestive signals, so things that may or may not be biology. It's really hard to get a definitive answer with some of these field-based assays. Oftentimes that requires taking samples back to the laboratory back home as well. But the goal is to optimize these assays and establish an order of operations based on incubation time, cost. Often disposable, consumable assays require significant funds, and resolution, like how much information did they give us? And hopefully that'll make more sense as I go along. The reason why we want to establish this order of operations is because in a mission scenario, it might inform where do you pause drilling and collect a larger sample, like did we hit a target of interest, and things of that nature. So some examples of the things that we measured for were cells, so the actual presence of living cells. And there's some fast assays that we used, like this kit called Petri films, but it's basically like a mini Petri dish on a piece of paper. And we directly applied meltwater and then looked for the appearance of microbial colonies. A microbial colony is just a mound of cells that originated from a single cell, so you can use it as a way to count how many organisms may be there. But it's very selective, right, based on what they're able to grow on. These are great. The problem is, what temperature do you incubate them at and for how long? And so this assay won't give you information within minutes. It really takes days. Other assays that we used to look for metabolic activity were what we called a Resagerin assay. And this is a redox dye that serves as an intermediate electron acceptor. And so it's part of central metabolism that uses an electron transport chain. And so it's an indicator molecule that changes color and would tell us if there's organisms actively breathing, basically. And this is a really sensitive assay. It's great, but it takes about 24 hours to see results. Faster assays, we did have these endotoxin assays. Unfortunately, these are fabulous. They work great, but they're really expensive. Endotoxin is a structural component on the outer membrane. So the outer part of the cell, you can think of it as like little appendages coming off the microorganism. They're present on gram-negative bacteria, and they're pretty common. So not all microbes, but this is a cellular component you might expect. And this is fantastic. It takes about a minute, but these cartridges are really expensive. And finally, likely biomolecules. This is an energy currency that all life uses. It's called adenine triphosphate, ATP. And we have two types of assays, one that we can do really quick that can happen in like seconds, and one that takes about minutes. So this is a good one. It's not definitive, but it's a pretty powerful assay that we've been using quite regularly to look for the presence of microbial cells. And we use this to clean all the instruments that we use. We test to see if there's any of these molecules present before we deploy the probe, and in background solutions like in purified water. And so it was really fundamental that we clean the exterior interior of this diver. That was a big portion of our work. This is for both code of conduct, but also cell numbers are really low in these environments. So for sample integrity, and in an analog scenario, it's also a fundamental part of planetary protection, which falls under the International Space Treaty. So a little bit on borehole structure. I'm sure somebody has or later will show you some footage from the borehole that was drilled with the ice diver during this project. But you can see as we go down in depth, the borehole structure changes. You can visibly see layers. And below on this bottom part are aliquots, about 30 mils of sample from each depth that we collected a discrete sample. And you can see that there's some changes in turbidity over depth, which is pretty cool. We weren't sure if we were going to see that level of resolution. And these visual observations were supported with measurements we made at the surface of silicate and just general absorbance. This green line here is our purified water standard. And so you can see as we got to the bed of the ice, it got more turbid. So this is in that subglacial reservoir. Microbial biosignatures seem to track with this turbidity that we saw as well, which makes sense. Microbes like to associate with particles. And so this is that endotoxin assay I was talking about. You could see it kind of peaks and correlates with some of the turbidity that we were seeing. This isn't graphed per depth yet. Again, this is hot off the presses, but this is direct counts. So this is a different type of measurement, just counting cells. The main take home here is that cells existed throughout the borehole. It varied with depth, often where there was more turbidity. But you can see these numbers are really low and actually pretty close to what you might measure in background water. These are at the limits of detection of some of these cell numbers. A lot of glacier melt is really pure. So that's just one of the challenges that we had. Um, Same measurements with depth with ATP, as well as these Petrifilm colonies that we were measuring. And I'm just kind of excited about this data because I don't know of a profile through a borehole taking measurements in real time of melt before for biology. So I think this was a really cool first that the Orca and JERP team was able to pull off. And so just trying to link it back to astrobiology, this is what Olivia is focusing on for her first manuscript. But NASA put forth these ideas like, how do you know if what you detect on another planet is life, right? Like I just showed you, oh, look, we measured ATP. Well, where does ATP fall? And it's actually pretty low. It's like this, down here, this potential biomolecule component, and if you just found that, in a sample from Europa, you might be excited, but you wouldn't have a press release necessarily. And maybe you would, I don't know, but you wouldn't claim for sure that you found life. You would need more definitive proof like growth or reproduction or metabolism or maybe multiple lines of evidence. And so that is what we were attempting to do with this project is to target assays that could give us multiple lines of evidence in the field and back home in the lab. And so that's it. I'll take some questions, but I just wanted to thank everybody and thank you all for the opportunity to talk about this. And yeah, I don't know. Should I stop sharing my screen or should I leave up the thank you slide? I don't know.
Thanks, Joe. Yeah, our camera's frozen in here, but we can still see and hear you. Figure that out in a second.
Oh, did you not see any of the slides?
No, we saw them all.
Oh, okay.
Just for some reason, it's frozen on the screen, like
Yeah.
the East Strand one.
But we can see you.
We can see it all. It's great.
Okay, great. Great.
Yeah, Esther.
Necessary, yeah.
Hi, thank you for a really fascinating presentation. I was wondering with the cryobots, you talked about their sort of dual capability of being used to send to other planets, but also doing research here on ours. And I was wondering what their capabilities of measurement are. Like once you send the probe down, what is it capable of?
Yeah. That's a fantastic question, and that's what the payload is. It's whatever you think you can send down a borehole, right? So I know that Dale Weinbrenner, the lead on this, has sent down like distributed temperature sensors. Um, Visions of Ice Diver are either planned or in use for projects like Coldex down in Antarctica, where you can send a smaller, faster version of a diver down to the base. Maybe it's a one-way trip in environments where you can leave equipment down there. And I think any type of tool that is, or instrument that is compatible with going on an instrument that heats up, has some electrical current and can move to the bed is an opportunity for you to make the measurements that you want. So I really see its potential in geophysics to maybe more rapidly and broadly deploy sensors at the bed. I'm fascinated with its ability to very cleanly, much cleaner than hot water drilling, get to the bed and collect some sample. It may not be able to collect as large a volume, or you might not be able to deploy as many instruments as you can down a 60 centimeter borehole. But I just think about the potential of more boreholes rather than just one big one that costs $10 million. Also, you can stick a cryobot in a sling load. So, yeah, I don't know if that, hopefully that gets at your question a little bit. Whatever you can dream up.
So Jill, can you hear me?
Yes.
Yeah, great presentation. So based upon the assumption that there's more life in a subglacial lake. So what we're seeing from your results, does that support the hypothesis that there's a subglacial lake?
I'm gonna leave that to Seth and Renee to decide what we were in. Um, I would argue I don't think that necessarily there's more life in a subglacial lake, probably than in the overlying glacial ice. But if the glacial subglacial lake is large enough, it could be like a really open water body. And microbes like nutrients. So sediment water interface, saturated sediments, swampy, soupy sediments, that's where I think there's a lot more microbial life, microbial activity, things that are really happening. So, but back to our point, it was really turbid, the basal reservoir samples that we collected. And so it does track that there was more microbes and more activity in those lower sediments. It was still low, and on par with some of the depths that had high sediment load. And so I think we have to do more analyses, but my question would be is, how often is this subglacial system scoured? Is there the potential for soft sediment and other materials and microbes to accumulate down there? I think that's still an outstanding question, but yeah, it's lending towards that. Like, and the data shows there's more in the bottom depths at least than in like the surface ice where it was like zero.
Yeah, well, like one thing that really caught my eye was that the turbid layers started, from 250 meters down. So,
No, there was some turbidity, I think, up in like the late 100s, early 200s.
or was it 270? Well,
Oh God.
it was quite high above the bed, and I was thinking, if
Yeah.
that ice is solid, how did that turbidity get up that high above the bed?
Well, I think Martin Trufer, I don't know if he's here, he should feel good about drinking the water no matter what Twitter says about him. But the layers, he was talking about how, when you see bare ice up there, right? When there's not all that snow, you melt, you accumulate a big sediment layer, right? And then if you have accumulation on top. So I think that the cells are associated with historic sediment layers that form in this basin. Again, I don't know as much about the glaciology of this system, but yeah. And to be fair, I think we missed the first 50 meters. I definitely expected to see a spike in maybe last year's snow that was left behind of any snow algae and sediment and stuff, but we didn't have great resolution. We were getting the kinks out those first 50 meters.
Yeah, it's great.
Thanks.
I can chime in real quick about this coming summer plant season. I'm not sure. Jess, you're gonna talk at all about what we're gonna do this summer? Okay, great. Never mind. I won't chime in. That's it covered.
All right, thanks, Jill.
Yeah, thank you.